Welcome to part 2 of the video on the PCR and cloning practical. We will now continue the practical with part 3 of the experiment, agarose gel electrophoresis and PCR product visualization. These are the reagents and equipment needed. Do check if you have everything before you start. Firstly, mix 10 microliters of each PCR reaction from the previous experiment that was resuspended in water with 2 microliters of loading dye. The mixing can be done either in an Eppendorf tube or on a piece of parafilm. Next, load the first well of agarose gel with 10 microliters of 100 base pair DNA ladder. Following this, load 10 microliters of each PCR reaction sample into the remaining wells. Make sure the sample is loaded directly into the wells and not into the running solution. Run the gel at 100 volts for 50 minutes. Do connect the wires in the right manner, red to red, black to black. For the next 50 minutes, the samples will be running downwards through the gel. Do check on the gel occasionally to ensure that the samples do not overrun. Next, transfer the gel to the gel documentation system. The teaching assistant will help you take a photo of the gel using the software. Do remember to take a photo of your gel using your camera. In part 4 of this experiment, we will be carrying out topo-vector ligation and transformation. Here, we will be cloning in the PCR products from the earlier experiment into the topo-vector. These are the reagents and equipment needed. Do check if you have everything before you start. Firstly, ensure that the topo-vector is kept in ice at all times. Next, label 2 1.5 ml centrifuge tubes as 1B and 2T. Now, add the components in the specific order as provided in the protocol. Flick the tubes with your fingers to mix the reagents. Perform a short 5-second spin to bring the components down to the bottom of the tube. Following that, begin the ligation process by incubating the samples at room temperature for 30 minutes. Place the tubes on ice while preparing for the transformation procedure. Collect 2 vials of competent E. coli cells and immediately place them on ice. Label the tubes 1B and 2T for identification purposes. Transfer 2 microliters of the ligated topo-vector from tube 1B to the corresponding tube 1B that holds the competent E. coli cells. Do the same for tube 2T. Mix the samples by gently flicking them. Do not mix the tube by pipetting. After mixing, incubate the tubes on ice for 30 minutes. Make sure the tubes are fully placed in the ice. After the incubation, heat shock the samples for 90 seconds in a 42 degrees Celsius water bath. Do ensure that the bottom of the tubes are completely submerged. It is very important to keep track of the timing during this heat shock process for a proper transformation. At the end of the 90 seconds, immediately transfer the sample onto ice. Next, add 250 microliter of LB to both tubes. Remember to use a fresh tip for each tube to prevent contamination. After adding the LB, place the tubes in a shaker and shake them at 200 rpm at 37 degrees Celsius for an hour. Make sure the tubes are kept tightly to prevent any spillage. At the end of 1 hour, take the tubes out from the shaker and prepare for the plating process. Spread 40 microliters of the transformed cell onto the labeled agar plates. Remember to use a fresh tip for each plate and work near a flame. Use a L-shaped spreader to spread the cells onto the plate in a circular motion. Remember to use a new spreader for each plate and work near a flame. Lastly, incubate the plates at 37 degrees Celsius for 16 to 18 hours. In the last part of this experiment, we will be counting the number of colonies on the plate. Collect your plates from the previous experiment and count the number of white and blue colonies obtained on each plate. Thank you for watching. Have a good laboratory session!